We want to excavate the treasures of our novel biomimetic in vitro test methods and make them fit for use in pharmaceutical industry. With this, I hand over to my young colleague and bench scientist Mikkel.
Thank you for handing me the word, Annette. I want to talk about a fundamental paradox in the modern drug discovery. We are incredible at designing highly specific molecules that bind tightly and unlock previously undruggable targets. That's the top half of the screen here. But this also comes at a steep cost. To achieve that specificity, over 70% of our newest pipeline molecules are extremely lipophilic and have near zero aqueous solubility, which then makes the oral absorption incredibly difficult, and we have essentially built a double-edged sword. Because of this extreme lipophilicity, our current drug development pipeline is fundamentally broken. When we test these complex, promising molecules or their advanced nanotechnological formulations, we rely on conventional static in vitro tests, essentially just dropping them in a beaker. And these tests fail to represent the dynamic human physiology, obviously. This then leads to massive false negatives, where millions of efficacious therapies are discontinued simply because our assays couldn't handle them. Worse yet, flawed animal models leads to false positives, and these then advance to phase 1 human clinical trials. They fail to deliver the therapeutic exposure in humans and force an expensive multi-year scramble to rescue the drug formulations. And we are losing time, money, and most importantly, viable therapies for the patients. This is where EXCAVATE changes the paradigm. We are proposing a revised predictive pipeline. In stage 2, we will replace the static beaker with a physiologically representative in vitro test method, where we simulate the dynamic conditions of the stomach, the intestine, and the gallbladder simultaneously. And crucially, our advanced time-resolved ultrafiltration technique actually only samples the truly absorbable fraction of the drug. And by integrating the nanotechnological formulation strategies early on and using our precise sampling method, we generate data that correlates 1-to-1 with the human plasma exposure. And we then feed this into advanced physics-based pharmacokinetic modeling, and the results are that we bypass the trial and error clinical scrambling, and we also deliver a de-risked, optimized therapeutic profile directly to phase 1 human clinical trials, speeding the development to patient process tremendously. Also, we potentially rescue viable therapies from the graveyard. And this is not just wishful thinking, we actually know this works. Here on the screen we see data from Lynnerup and Holcim, which are our published proof of concept. The graph on the left shows that our technique vastly outperforms conventional approaches in predicting the human absorptions. And the graph to the right shows that we can even feed this into set pharmacokinetic models and also get superior human plasma concentration predictions. We already partnered with the University of Athens, who are leading experts in advanced in vitro performance testing, and we want to test on a larger library of compounds and drug formulations. To turn this proven concept into a global standard, we are building an open access, pre-competitive tool, but we need your help to stress test it. We seek pharma and biotech partners to provide the human pharmacokinetic datasets and formulations across diverse disease models to challenge our platforms, as well as aiding in the physics-based pharmacokinetic modeling department. Our ultimate vision is simple. We want to establish an open, pre-competitive in vitro tool that closes the translational gap, optimizes the clinical pipelines, and enhances the patient's safety.